Hello friends, today I am going to explain you chapter 12 Biotechnology and its application, part 1, class 12 biology, subject biology based on NCERT. But before explaining, I would like to like, share and subscribe our YouTube channel so that you may always get an update of new videos. Biotechnology actually engaged in the producing genetically modified organism, microbes, fungi, plants, and animals for industrial scale extraction of biopharmaceuticals and biologicals and in agriculture. There are basically three critical areas of research like catalyst, searching and providing best catalyst in the form of improved organism, genetically modified microbes or pure enzyme, and for that optimum condition is also required. The catalyst is provided with sterilized medium, sterilized bioreactor, proper pH values, optimum temperature, aeration and regular stirring for maintaining it at its physiological most active state. Downstreaming processing. What is downstreaming processing? It involves recovery of product, its purification, processing and formulations. Biotechnological applications in the agriculture. There are basically three options to increase the food production. How we have to increase the food production for the huge population? Nowadays agricultural land area is also going to be decreases, so that there are three basically options to increase the food production. Agrochemical based agriculture. In this we have to use the fertilizer and chemicals. Organic agriculture, genetically engineered crop based agriculture. So there are three basic areas. The green revolution succeeded in increasing the food production, but it was not sufficient to feed the growing human population. Actually, green revolution is a tremendous increase in the food production, but it is very difficult to feed the growing human population. Increased yield is partly due to use of improved crop varieties. However, its reason are better management practices and use of agrochemicals such as fertilizers and pesticides. But due to the higher cost of agrochemicals, the farmers of developing countries are unable to afford. Another effect of agrochemical is that very harmful for the human population as well as our ecosystem. If you are putting fertilizers and chemicals in the field that are flowing away from nearest lakes and ponds, that disturb the ecosystem, because that is very harmful, that are accumulated in the body of the organisms also. We should minimize the use of fertilizers and chemicals so that their harmful effect on the environment also reduced. One solution of the problem is the use of genetically modified crops. Genes of plant, bacteria, fungi and animals have been changed by manipulation. Therefore, these organisms are called genetically modified crops. The behavior of genetically modified crops or organism depends on the nature of genes transferred, nature of host plant, bacterium and animal. Norman E. Borlaug, the full name is Norman Ernest Borlaug, the father of Green Revolution, developed a high yielding short straw and disease resistant wheat. So that for the development of this variety of wheat, tremendous increase in the production of wheat, he got Nobel Peace Prize in 1970 for his contribution towards increase in the production of wheat. MS Swaminathan is considered the father of Green Revolution in India. Now we have to discuss about the genetically modified crops. GM crops are transgenic crops which contain and express one or more useful foreign genes. The technique of GM crops has two advantages. Any gene from any organism or a synthetic gene can be incorporated. Change in genotype is precisely controlled, and production of transgenic plant, it is based on TI plasmid. I had already discussed in the previous chapter the plasmid is a extra chromosomal circular DNA of the bacteria. But here we have to discuss about the TI plasmid. TI, the full form is tumor inducing plasmid, which occurs in the soil bacterium Agrobacterium tumefaciens. The name of the bacteria is Agrobacterium tumefaciens. The bacterium is called natural genetic engineer. Basically, it causes the crown gall disease in the plants because genes carried by its plasmid produce effect in several parts of the plant. It induces formation of cancerous growth. That crown gall disease or the crown gall tumor is a cancerous growth in the plants. This transformation of plant cell is due to the effect of TI plasmid. TI plasmid means that plasmid that consists of the gene tumor inducing plasmid carried by the pathogenic bacterium. Hence, for genetic engineering purposes, agrobacterium strains are developed in which tumor forming genes are deleted. It means that is one of the most important part. The tumor inducing genes are deleted. These transformed bacteria can still infect plant cell. That capability of the bacteria is not disturbed. Only we have to delete the genes which are causing tumor in the plant so that these transformed bacteria can still infect plant cells. The path of TI plasmid transferred into plant cell DNA is called the tDNA. Such plants cells are cultured, induced to multiply and differentiate to form plantlets. Another transgenic plant we have to discuss here, that is BT cotton. What is BT cotton? BT, the full form of BT is Bacillus thuringiensis. Bacillus thuringiensis, and this is a bacterium, produce proteins that kill certain insect like lepidopterans, coleopterans, and dipterans belong to different insects. That BT toxin protein exists as inactive protoxin. But once the insect ingests the inactive toxin, it is converted into an active form of toxin. Due to alkaline pH of the alimentary canal of the insect that solubilize the crystals, the activated toxin bind to the surface of midgut of epithelial cells and create pores which cause cell swelling and lysis and finally cause death of the insect. The toxin is coded by a gene named CRY. DNA is responsible for the protein, toxin protein, and that gene, small part of DNA named CRY, the CRY, that is also known as CRY protein, the protein which encoded by the genes CRY1AC and CRY2AB controls the cotton bollworm, and cotton bollworm infect the cotton plant, so that we have to introduce this gene into the plant when the insect are sucking the sap of the plant, so the epithelial cells of the gut of the insects are dissolved and the insects are died. So in this way we have to save the crop, and this variety of plant is known as transgenic plant. Now we have to discuss about the pest resistant plant. The nematode Mileidogyne icognitia affect the root of tobacco plant and causes great losses in yield. And one of the technique, that is RNA interference, is the process on which a strategy has been developed to prevent the infestation. What is RNA interference? This method involves the silencing of a specific messenger RNA by a complementary double-stranded RNA molecule that binds to and prevent translation of messenger RNA. The source of complementary RNA could be form of infection of viruses having RNA genome or mobile genetic element called transposons. Mobile genetic elements are called transposons that replicate via an RNA intermediate using agrobacterium vector. The nematode specific genes were introduced into the host plant. The introduction of DNA was such that it produces both sense and antisense RNA. It means DNA is producing the RNA, so that this DNA of nematode is producing sense and antisense RNA in the host plant, in the host cells. These two RNA being complementary to each other, forming as a double-stranded RNA that initiated RNA interference, so that RNA interference and silence the RNA of the nematode. Hence, the parasite could not survive in the transgenic plant. So, in this way, we have to uh, save the crops, and these are the methods we have to use in the biotechnology to save the crops. That is a diagram of cotton bollworm and destroyed by bollworm that is a fully mature. And after that we have to discuss about the some questions related to this part, that is crystals of bt toxin produced by some bacteria do not kill the bacteria themselves, because the first option is bacteria are resistant to the toxin, toxin is immature, toxin is inactive, bacteria encloses toxin in a special sac. That is a mcq type question. And what are the transgenic bacteria? Transgenic bacteria, it means that I had discussed. Illustrate using any one example. Compare and contrast the advantage and disadvantage of production of genetically modified crops. What are cry proteins? Name an organism that produce it. How has man exploited this protein to his benefit? So these are the questions we have to discuss here. You have to do at your home. And this is the part one of this chapter. uh, Rest part we have to discuss in next videos. Thank you, students. Have a nice day.